In this video, we're looking at DNA replication. We're looking at how DNA copies itself to turn one cell into two cells, and each one of those cells being identical to each other. So we're copying the entire library, the entire genome of the cell to make an exact copy of it. So this is a copy of the entire genome, DNA replication. So we're copying DNA. DNA, if you remember, is anti-parallel, meaning one strand is running from a 3' direction to the 5' direction. It's anti-parallel, so just like in a highway or a street, when you have traffic going in one direction, you're going to have traffic on another road that's parallel running in the opposite direction, the anti-direction. The other strand is the opposite, is the anti, running from the 5 to 3 as opposed to the 3 to 5 on the top. So because of this, when DNA replicates, when DNA copies itself, it's going to copy itself in opposite directions. So I'm looking at the DNA strand, and you see the blue dotted lines. Those are the hydrogen bonds that go between the nucleic bases. Adenine will always go with thymine, and thymine will always go with adenine. So is cytosine and guanine. They always pair up in those particular combinations. And then you have the outer edges where you have the alternating phosphate and sugar group. Sugar, phosphate. And those bonds that keep those together are called covalent bonds, which are slightly stronger than your hydrogen bonds. All right, so again, here are the different enzymes that are at work in DNA replication. So you have the DNA molecule, obviously, helicase, DNA polymerase, RNA primase, DNA ligase at the end. So one of the things that I didn't mention a second ago is that topoisomerase will be the first enzyme to show up, and it will unhelix DNA. Remember, DNA is like a ladder that is twisted. So topoisomerase will show up, unwind it, make it back into a two-dimensional plane, and then helicase will start breaking those hydrogen bonds, separating the two strands. These two strands are called the parent strands, the top one and the bottom one. As helicase breaks up those hydrogen bonds, that particular location where those bonds are being broken is called the replication fork. Now, one of the things that we want to consider as we're moving forward, let's take a look at the top strand, for example, the top parent strand. Notice the configuration of the deoxyribose sugar. You have the 3' on the left, and then you have the 5' on the right. So the parent strand on the top is running from the 3' to the 5'. This is really important, because at the bottom, that means it's running the opposite. That means the 5' end is on the left and the 3 is on the right. This is important when we start to consider how DNA is actually being replicated, the direction that it's being replicated. So at the top, our exposed end is the 3' end. And at the bottom, our exposed end is the 5' end, as I mentioned before. So that means when I start laying down these new nucleic bases, the new strand will be copied in the 5' to 3' direction from the perspective of the new strand now. So as I lay one after another nucleic base, it is being laid down from the 5' to the 3'. And it's going to copy towards the replication fork. So DNA polymerase, this enzyme, this third enzyme, is the one that actually lays down the nucleic bases. But before I continue, understand that DNA polymerase needs a starting point, and it's the RNA primase that's the one that lays it down. Some books refer to it as DNA primase, so just keep that in mind. Just remember that it's the primase that lays down the RNA primer to start off the DNA polymerase. And it copies in the same direction that the helicase is opening up. It's the same direction that the replication fork is going. So it is able to copy and lay down nucleic bases continuously. This is the leading strand. Now let's consider the lagging strand, the one at the bottom. Okay, it has to copy in the opposite direction because it needs an exposed 3' end. So what will happen is that the RNA primase will come down, it'll lay down the primer, and that tells the RNA or the DNA polymerase where to start copying. So the RNA primer lays it down, and now DNA polymerase will show up and start laying down its nucleic bases in the opposite direction in fragments called Okazaki fragments. So finally, we are almost done. The only issue is that we still have RNA attached to DNA. These RNA primers need to be removed. These starting lines need to be removed. And that is where DNA ligase shows up. DNA ligase is the enzyme that will replace the RNA primer with DNA, and it will solidify, it will close up any covalent gaps that are in the strand, in the new strand. So they seal up, they glue those covalent bonds, allowing phosphate and sugar to recombine, to shake hands again, creating a stable DNA molecule. And it does the same for the top one as well. The only difference is at the top one, you only have one primer to get rid of, whereas at the bottom, you have several primers to get rid of in the Okazaki fragments. So you end up with two DNA molecules, two brand new DNA molecules that will then become a brand new cell, each on their own. And if you consider both of these new strands, each of these new strands each of them have part of the parent strand, the original strand, and then the other half is the newly copied DNA bases. This is what we call semi-conservative replication, where both strands, each of them have half original and half new. That is semi-conservative replication. All right, guys, thanks for watching, and I will post some more videos to help you on this subject. Thank you.